Okay, so as we continue with identification of microorganisms, I already alluded to the enzymatic activity, how it also is being used to help differentiate certain microorganisms from one from another. So many tests utilize these types of techniques in determination. We already talked about fermentation and how it was specific for carbohydrates in these studies. The use of selective and differential medians, you've already gone over from the previous study. We already talked about the numerical classifications that are used to be able to identify particular constituents of various microorganisms. This just gives you an example of a numerical system that's helped to be able that helps in being able to identify particular types of microorganisms. Serology is also a study that's used. It combines a particular antibodies or antigens that are in serums of particular organisms. You can use a comparison of what you've seen from historical studies that have been done, use that in comparison to what's found in studies that you're currently running on microorganisms. That comparison helps you to identify a particular microorganisms. We'll talk about agglutination in just a bit, but certain microorganisms will secrete substances that will help red blood cells and platelets to kind of clump together. So the ability of certain organisms to cause agglutination, the clumping to occur in particular cells, helps to identify certain microorganisms or the substances these microorganisms may secrete from others. ELISA is another in the Western blot. We talked about the Southern blot. These are just related studies that help, again, to identify particular microorganisms based on the serology in these substances. Your book goes through the steps of Western blot, how it helps to identify the organism in Lyme's disease, Borrelia. And you can read through those steps in your reading. I won't go through each of the steps now. Basically what I would like you to know about Western blot, Southern blot, ELISA, these are just types of studies, again, to help identify particular microorganisms. Remember this slide. I'm going to talk about phage typing right now, but remember this slide because many times there are so many microorganisms that are trying to be identified under a microscope, and it's not possible to look at each individual organism, nor is it possible to count the total number of microorganisms that are under a microscope. And so typically these slides are placed under grids. The grids are actually on the microscopic lens. And so when the technician or a scientist is looking at a particular type of organism, and again, this is just to determine the number of microorganisms that are on a cultured plate, they may report the number of microorganisms per high-powered field. This is a field, one of these grids. And you basically get the number of microorganisms that are in a particular field. You plug that number into a formula. It gives you the total number, or what would be an estimated number at best, in the whole culture median. Based on that number, we can determine if there is an adequate amount of bacteria to actually cause disease. A urinalysis, for example, if we're checking that for microorganisms, urine may have bacteria. Urine is a great medium for bacteria to grow. Though it's the waste from the human body, it's nutrients for certain microorganisms. And so a small number of microorganisms found in the urine. Now, urine should be sterile. You shouldn't see microorganisms in the urine. But below a certain count, below 100,000 organisms per high-powered field, it's not considered to be a major urinary tract infection, and typically we won't treat that with antibiotic therapy. It has to be over a certain number, and I'm just showing you this picture because it gives you a good idea of how the grids are used to help determine the high-powered field. Now, phages basically bacterial viruses, as we talked about in the previous slide, and again, phage typing is commonly used to help track particular microorganisms. There are commercial systems that also help identify organisms based on the fat content. Again, looking at the chemical constituents, flow cytometry, we already went over in the previous portion of the lecture. Flow cytometry uses differences in electrical conductivity. These organisms conduct electrical activity differently. They weight differently. They also will reflect and or absorb light differently. So fluorescein in some of the species is an important constituent. Some of the cells will selectively stain for particular antibodies based upon fluorescent dyes. But this is the technique that illustrates and utilizes flow cytometry. Again, the steps of flow cytometry, the use of the laser beam or the light beams, is described more in your textbook, and please read that as a part of your reading. We also talked about the base pairing compositions and being able to identify particular microorganisms. The base pairing obviously is a part of the genetics of a particular microorganism. And so because of the base pairing, you're able to determine the percent composition of particular base pairing. Knowing the composition of the base pairing helps you to identify what type of organism may be involved or you may be trying to identify. Again, this base pairing gives the DNA fingerprint, as I alluded to earlier, through a process called electrophoresis of restriction enzymes. These restriction enzymes, and remember what the restriction enzyme does. It goes in, it cuts out a particular portion of the DNA sequencing. Cutting out that particular portion of the DNA sequencing helps you to be able to identify particular microorganisms. So, in this illustration out of your textbook, not illustration, this picture out of your illustration, out of your textbook, just demonstrates how this particular pattern. And actually, if you can get the pattern of one microorganism, you can line that with another microorganism if they have similar patterns. And this is how they identify, you know, who the baby belongs to. This same technique, based upon the base sequencing of the DNA, helps to identify. If it perfectly matches up, then you know there is a significant relatedness between the microorganisms, or a specific relatedness between the child and the parent and the offspring. And again, we already talked about restriction enzymes. We already talked about the polymerase chain. Nucleic acid hybridization as well, when we divide or when you allow the DNA segments to be heated. Remember, we used heat. We can use a change in pH that will break apart or separate the hydrogen bonds of the base pairing. And then you can use this information to identify, or you can use it in a technique of hybridization to put two organisms together, or two base chains together. And if they bind, if the base sequences of those organisms bind, it will determine a relatedness between organisms. This organism have DNA structures. You divide this DNA structure, you divide this double helix on this side, you put it all in the same soup, and you mix it around, and these complementary base pairs from different organisms will bind to each other. And it demonstrates a relatedness. Why? Because the base pairing sequence, remember, will only bind to similar complementary pairs. And so if the complementary base strands will bind to another microorganism, again, it gives you the appearance of relatedness. And you've used now a technique of hybridization. The technique assumes that two species are similar or related, and so major portions of the nucleic acid sequence will also be similar. Again, another identification method. Southern blotting, we already talked about. This is the nucleic hybridization, and again, you can go through these steps, read through these steps in your reading material. Again, this just kind of describes the dichotomous key that I referred to again, information concerning points of microbial identification placed into a dichotomous key, where workers will again answer a successive number of questions, each one leading to a next step and the next step until you're able to identify a microorganism. The cladogram is basically the map which identifies the evolutionary relationship. You just need to know that definition. Again, each branch point is defined by features, and you'll see there's RNA sequences that are used to help make genetic information, help to make up the cladogram, or the information that's stored there. This is an example of a dichotomous key. Like some of the questions that would be answered, you move down the step to be able to help identify a particular organism. You can start with a gram stain reaction. Is it negative? Is it positive? If it's a negative gram stain, then you look at what's the metabolism that's typically related to gram-negative organisms. Glucose fermentation, acid or gas produced, just acid produced, it leads you to a different type of organism, specific organism that produces acid in this type of step. If it produces acid and gas, then there are a series of other questions that you have to ask. You'll find out, is urea being hydrolyzed in this process? If not, or if it's not being utilized, then it moves you down through another step. If it is, then you quickly identify a particular type of microorganism. If not, you continue to move down and continue to ask questions until eventually you get to a point where you can identify specific microorganisms. Now, I'm not asking you to be able to go through these steps and to recognize each of these organisms or to identify whether or not particular chemical substances are produced or hydrolyzed. What I want you to appreciate is the steps of the dichotomous key. This is what the laboratory person would go through to be able to identify a particular organism. There are particular steps in this process. So when you send a specimen down to the lab, you may be able to get a gram stain right away, but to be able to identify a particular organism within that first hour of time is not always possible, because a sequence of steps have to be done. Particular chemical studies may have to be done, biochemical assays, checking particular types of enzymes that are produced by this microorganism lead to the identification of the organism specifically. Sometimes these studies have to be done in specific labs. A small hospital, for example, is great in being able to do a gram stain, would be great in being able to identify gram positive from gram negative, could culture out on a regular median, selective median nutrient auger would be able to identify a common organism. But if it requires identifying particular enzymes to be able to identify that organism, every lab may not be capable of running those studies. So at a certain point, to finish the dichotomous key, let's say you determine that urea was not hydrolyzed in this particular organism, then you may not be able to do the next step, and that specimen may have to be sent out. And so a lot of times you'll call the lab and say, hey, did you identify that organism? You say, no, we had to send it out. Well, it lets you know somewhere in this processing it was beyond the capability of that lab. Not that that's inappropriate, but if you only have to identify one of those organisms per year or every 6 months in your lab, then it's not cost effective to keep the equipment that's required to do some of the biochemical assays that may be required. So then it goes to a reference lab, and the reference lab would then be the system to be able to take to identify the organism further. But all of these are the steps in being able to identify an organism. Again, a sequence of steps, a sequence of questions that have to be asked, which will eventually lead to the name, the identification of a particular microorganism. Now, we use a virtual lab, but in some, if you study microbiology as a major, you would have to perform a series of lab tests to be able to identify a particular microorganism. You may be started off with Klebsiella pneumonia, but you start off by doing your gram stain first and walking all the way through these steps, and as you answer yes or no to each of the questions, it would eventually lead you to identification of Klebsiella. Same for Yersinia and Pasteurella organisms as well. The cladogram, again, is the information determines the sequence of RNA-based pairing. Again, the construct of that you can please get in